Hello everyone, myself Dr. Geeta Chaudhary from S.S.Jain Subodh PG College, Jaipur, Rajasthan. My today's lecture topic is gene cloning. First of all, we have to understand what is the gene, where we can find this. Then what is gene? Gene is the smallest unit of cell. This gene found in our tissues. Tissue contained of many cells, and cells is a structural and functional unit of life, in which cells contain a small part of this cell is nucleus. Then this nucleus contains various genes that are involved or are responsible for the characteristics that carry the general characteristics from one generation to other generation. In this slide you can see the structure of gene, and this whole gene have different or more than thousand or more than lakhs of chromosomes. And what we can say, the gene is basic physical and functional unit of hereditary, and genes, which are made up of DNA, act as instructions of molecule called proteins. You can see the structure of chromosomes, and this chromosome is called the DNA. This is deoxyribonucleic acid. Then we will discuss about the various steps involved in gene cloning. First, we have to uh, know what is the cloning. Cloning is a process in which we generating a identical copy of a cell or of any organisms. Clones are those organisms that have identical to the genes. Then there are many steps involved in gene cloning. First step is, first we have to identify the gene to be cloned. Then we have to isolate that gene from the specific individual cells. Then we will take this gene, then transfer this gene to the specific vector that are the vehicle. We can say vectors is a work as a vehicle. Then first in these steps of gene cloning, we have to generate the DNA molecules. For this, we have identified a specific species or specific individuals that they want their gene to be cloned. Then we have to isolate that gene. Then after the isolation, we have to cut a small fragment of that gene from the whole genome, because large amount of gene or large size of DNA molecule or large size of DNA fragments cannot be cloned. That's why we have to cut a small amount of DNA molecules. We use different type of enzymes to cut the DNA molecules. These enzymes are, for example, nucleases. We can use these enzymes, nuclease, to cut the DNA molecule. Then we have to take out these genes, and we transfer these genes to the suitable vectors. Then what we can say, and you can ask, what are the vectors? Vectors, or we can say these plasmids. What are the plasmids? Plasmids are extra chromosomal part of any bacterial cell, plant cell, or any yeast cell, fungus cells. And these extra chromosomal part have their ability to freely replicate in their host cell. So, in which we use this vector as a vehicle for the cloning. Then we have to select these suitable vectors to cloning. Then we insert this foreign DNA or DNA to be cloned into vector with the help of ligase enzyme. We join the DNA molecules to the vector. Then these vector containing DNA fragment is called a recombinant DNA, and then we have to introduce these recombinant DNA into the host cell. Then we transfer the host cell into the any cultured media, by which when bacterial cells or any host cell multiply or host cell divide, then the vectors or plasmids, they can also divide or they can also multiply. After the multiplication of host cell, we have to isolate these bacterial cells or plant cell in which we clone the DNA, or in which we transform the vectors containing DNA. Then we have to select the specific plasmids, or we have to screening these plasmids and isolate these recombinants, and we have to use for their any disease control or gene therapies. In this slide you can see the method, or you can see the steps, various steps involved in the gene cloning. One basic cloning technique begins with the insertion of foreign gene into the bacterial plasmid. Earlier we have discussed what are the plasmids. Plasmids are the extra chromosomal part of any bacterial cell, plant cell. They can live in plant cell or any bacterial cells. According to our requirement, we can choose this host cell for the plasmid isolation. So first of all, we have to choose this plasmids for the cloning. For example, we will take this bacteria cell for the plasmid isolation. Then we treat this bacterial cell for the enzyme, uh, by the enzymes for the isolation of plasmids. Okay. When we isolate these plasmids, then we have to select the genome to be cloned. So first we have to identify any cell or tissues in which we can isolate the DNA. Then we take out the gene from the any cell. Then isolation of plasmid and DNA, and DNA containing gene of interest, we mix with these DNA to the plasmids. We can use here a ligase enzyme to join these DNA fragments into the plasmids. Plasmids are the freely replicative extra chromosomal part and also have an origin of replication, and they also have antibiotic resistance genes, or they work as a screening. Then we have cut out the big fragment of this gene with the help of nuclease enzyme that insert into this plasmid or join in this plasmid with the help of ligase enzyme. This plasmid containing the gene to be cloned is called a recombinant DNA or recombinant plasmid, and this is the recombinant because this is made by the combination of plasmid and gene to be cloned or DNA fragment. In this next step, then we have to choose the host cell to replicate this plasmid. Then we transfer this plasmid, or we put this plasmid into the host cell. This is the bacterial cell. Then this bacterial cell is called a recombinant bacteria because they have the recombinant DNA molecule inside their cell. Then we have to prepare a culture media, and we can transfer these bacterial cells in these culture medias by which they can replicate, they can divide, or they multiply, and they form the different cells. You can see this in this slide. There are many number of cells. They contain all recombinant DNA inside their cell. Then these are the different number of plasmid and different number of chromosomes or different number of recombinant DNAs there inside the cell. Then we have to isolate these plasmids containing recombinant, containing gene to be cloned. Then we have to rupture these bacterial cell wall, then isolate from this bacterial cell, isolate the plasmids containing DNA, and that's why we can isolate these bacterial cells, we can isolate the plasmids from the bacterial cells, and we can use these genome or different number of copies of the specific gene to be cloned, and we can use these gene in the different disease treatment, gene therapy, cancer treatment, and other different diseases. This is my small video. I hope you liked it. Thank you so much.